Hello everyone, Kelton here. Today we are going to be discussing lipids and talking about fatty acids. So go ahead, read the outline, and we're going to jump right in. Lipids is kind of a fancy science term, so what's a general term that you've heard to describe a lipid before? And perfect, so the generic name for a lipid is fat. So as many of you know, fats are non-polar and hydrophobic, which means they don't mix well with water at all. Another general characteristic of fats is that they have long hydrocarbon tails. So could you use that name hydrocarbon to draw a hydrocarbon tail for me? And perfect. So as the name implies, a hydrocarbon tail is just a tail full of hydrogen and carbon, as we see here in this image. While many of us probably go to the gym to try to decrease the amount of lipids we may have, lipids do have a vital role in our bodies. So can you think of what lipids are used for? And great, so while fats might get a bad name, lipids are really important to us. Lipids are used for energy storage, they're used for insulation like the blubber on this walrus, and also they're utilized in cell membranes. Lipids are also used for hormone synthesis as well. Triglycerides, which we see here, are your typical lipid molecule. Triglycerides consist of one glycerol molecule, which is just an organic alcohol, and three fatty acid chains as we see here. This reaction shows our glycerol and our three fatty acids coming together to form a triglyceride. So what type of reaction does this? And perfect, so hopefully you keyed in on the fact that water was a byproduct and that multiple subunits were combining into one. This makes this a dehydration reaction. During this reaction, we see that an ester linkage is formed between the glycerol and each of the three fatty acids. You might recall that this reaction is how polysaccharides are formed from monosaccharides and how amino acids come together to form a polypeptide chain. So knowing that these reactions are similar to the ones we saw with our proteins and carbohydrates, what type of reaction is occurring here where we're breaking a triglyceride apart into a glycerol molecule and three fatty acids? Great, so this is a hydrolysis reaction, and remember, a great way to key in on this is that we're adding water and we're breaking something apart. Now we're going to talk about fatty acids a little bit more in depth. Fatty acids then are just organic molecules that contain a carboxylic acid functional group and a hydrocarbon tail. These fatty acids can vary in length, although most tend to be about 12 to 18 carbons long, and they also can vary in degrees of unsaturation. A degree of unsaturation, to put in simple terms, is just a double bond. So in this image, could you label which is the saturated fatty acid, and then could you also label what the other one is? So great, another way to think about this one is the saturated fatty acid is the one that's saturated with as many hydrogens as possible. We see that's the case with this one that has no double bonds. An unsaturated fatty acid then has a double bond. Because saturated fatty acids don't have any double bonds, they have a really straight configuration. This straight configuration allows them to pack really tightly together. So this tight packing means that they're a solid fat at room temperature. Can you think of any fats that are solid at room temperature? And great, so butter's a really good example of a saturated fat. There are multiple different types of unsaturated fatty acids we can have. If a fatty acid has one double bond, we call this a monounsaturated fatty acid, as we see on the left. If an unsaturated fatty acid has two or more double bonds, we call this a polyunsaturated fatty acid. A cis-unsaturated fatty acid occurs when we have our two groups bonded to the same side of the double bond. This results in a kink in the chain, as we can see here in this image. These kinks in the chain mean that these fatty acids are not capable of tightly grouping together and they leave gaps. This loose packing results in cis monounsaturated fatty acids being liquid at room temperature. Can you think of any fats that are liquid at room temperature that you... And perfect, olive oil is a great example of a cis unsaturated fatty acid. As opposed to a cis unsaturated fatty acid, trans unsaturated fatty acids have their groups attached to opposite sides of the double bond. Notice that this results in no kinks of the chain of a trans-unsaturated fatty acid, and their structure closely resembles that of a saturated fatty acid. This allows trans-unsaturated fatty acids to pack really tightly. What are some things that you maybe have heard about trans-unsaturated fatty acids before and where they come from? And great, so you probably remember hearing that trans fats are bad for you. Trans fats are made through man-made industrial hydrogenation, and we typically don't find very many trans fats in nature. Another common lipid that utilizes fatty acids is a phospholipid. Do you think you could label all of the parts of the phospholipid here? And nice, so hopefully in labeling this, you notice this looks kind of similar to a triglyceride, except in the place of one of the fatty acids, we have a phosphate group. We still have a glycerol, and we still have two fatty acid chains, but now we've just added a phosphate group onto this. So, what term do you think is going to describe the polarity of this phospholipid? Is it going to be polar? Is it going to be nonpolar? Or is it going to be somewhere in between? And great, so phospholipids are known to be amphipathic. This means that they have one side that is hydrophobic or water-fearing, that's going to be our fatty acid tails, and they have one side that is hydrophilic or water-loving, and that's going to be the side with the phosphate group on it. Another key trait of a phospholipid is that they spontaneously assemble into bilayers. So where do we find phospholipid bilayers? And nice, so hopefully remember phospholipids are associated with membranes. This could be the cell membrane or the membrane surrounding organelle. Okay, so now that we've finished with the lesson, let's go ahead and have a mini quiz. So, which of the following fats is a liquid at room temperature? So starting off with A saturated fat is something like butter. Butter is solid at room temperature, so we can discount this one. B is a trans-unsaturated fatty acid. Remember that trans-unsaturated fatty acids are capable of tightly packing together like a saturated fatty acid. This ability to be tightly packed results in trans-unsaturated fatty acids typically being a solid at room temperature, so we can get rid of this one too. Answer C is a little bit silly. It's a short-chain polypeptide. Remember, polypeptides are proteins and not fats, so we can get rid of this one as well. This leaves us with answer D. So a cis-unsaturated fatty acid is something like olive oil, which is a liquid at room temperature. So D is our correct answer. So now that we've finished with the lesson and had a quiz, let's go ahead and do a review to see what you learned today. What is the difference between a fatty acid and a triglyceride? Next, what are lipids used for in your body? And lastly, what happens if we try to mix olive oil with water? If you can answer these questions, you are well on your way to success. Everybody be sure to have a wonderful day, and I will see you in the next one.